All right, welcome everyone. We're so glad to have you joining us today for today's webinar from Insmed. I'm Leon Orlando, Cure Duchenne's VP of Research, and I'm going to get us started with a little bit of housekeeping. We're going to have the opportunity for question and answer at the end of the webinar. So at any time throughout the speaker's presentations, you can click on the Q&A button at the bottom of your screen. If you don't see the Q&A button, I think if you have your mouse down there, it will appear. We're going to be recording the webinar, and after today, it will be available on Cure Duchenne's website in case you would like to share it with others or to listen again. So with that out of the way, I just want to say it's such a pleasure to be hosting the Insmed webinar today. As a lot of you are probably aware, Cure Duchenne tries to keep research moving forward towards transformative treatments and cures. And one of the ways that we do this is occasionally making investments in companies that have promising technologies. And although we bring you webinars about all of the therapies in development and clinical trials going on in Duchenne, in some cases, we're talking with a company that we've made a past investment in, and that is the case today. So for full disclosure, Q Duchenne made an investment in Insmed in 2023, and that was after a few years of talking with their scientists about the potential of this differentiated delivery route that you're going to hear about today. And I'm so excited for you guys to learn more about it. So for that, I'd like to welcome Stephanie Duffy, Insmed's Head of Patient Advocacy Engagement in Gene Therapy, to the screen. Hi, Stephanie. Good to see
Hey,
you.
Liana, how are you?
Great.
Good.
I'm going to head off so you can take it over from here.
Perfect. Thank you. And hi, everyone. It's with early investments that Liana mentioned that really helped to drive science forward. So, with that, I'm Stephanie Duffy, as Liana mentioned, and I am the head of patient advocacy for our gene therapy portfolio at Insmed. And we are so excited and grateful to be here. As Liana mentioned, it's been some time coming to share our preclinical work and importantly, our first in-human clinical trial for gene therapy in Duchenne muscular dystrophy. But before we get started, it's important for those that aren't familiar with Insmed to really understand our mission. And what I want to draw your attention to is the center of patients and talking with patients first, listening to patients and caregivers and making sure that we always conduct our research with input in mind. And with the job that I get to do each and every day, I can speak to the fact that we do speak with the community and with our patient advocacy partners and healthcare providers as well to ensure that our protocol and all of our clinical development activity reflects what everyone is hopeful to see. And with that, we can advance over to the next slide. I've introduced myself, but I'd also love to share that I am a mommy of three. I know we have a lot of parents joining us here today to learn some additional information on behalf of your kiddos. And as a mom of three whose little one lives with a rare neuromuscular condition, we appreciate you being here and gathering that extra information on behalf of your family. And with that in mind, I want to welcome our partner at Insmed and key opinion leader, Dr. Panda, who probably needs no introduction as you're a very well-known pediatric neurologist studying and working in Duchenne muscular dystrophy at Arkansas Children's and also one of the investigators for Ascend, which we'll talk more about today. So with that, Dr. Panda, happy to hand it over to you. And thanks for being here.
Thank you, Stephanie. Thank you, Leanna and Deborah on the Duchenne for this. I hope you all hear me okay. Um, let's kind of get into this and into the Insmed, the gene therapy development. And I am being compensated for speaking today. I'm one of the investigators for the trial, that the first in human trial that's being done. All right, so just kind of briefly talk about what, we know the microdystrophin, we have been talking about this for over the years. So here, the INS1201 is also microdystrophin. They're using the AAV9 capsid to deliver this microdystrophin gene. Here is your microdystrophin gene construct that includes exons 1 to 17 and 59 to 69, and it has a promoter that is a muscle-specific promoter that, again, all of this to make so that this microdystrophin is being delivered to the muscle. And in the trial, it's given us a one-time intrathecal injection. Intrathecal meaning through spinal tap into your spinal space in the back. Like, when I usually explain this as like how moms get epidural during childbirth. That's kind of similar technology given intrathecally. So I think with this specific intrathecal route, it's being given one fixed dose, and it's not a weight-based dose. And this method is being studied to understand if it may help the treatment to reach the muscles and heart muscle as well as more efficiently with a lower dose. And again, like I said before, it is like an epidural, kind of similar to the epidural. So this kind of talk about a little bit about the preclinical studies, mainly studies in the animals, either mice or non-human primates. And this first here you look at the comparison between an intravenous delivery and an intrathecal delivery of a gene that expresses a muscle protein. And this is just to compare these two deliveries at the same doses. If you look at here, these are different muscles that's being studied, like biceps, triceps, deltoid, including heart and liver. And the top row up there is the non-injected. There's no protein or no gene that's being given. And then you have your intravenous through the vein, and then you have the intrathecal delivery. And I think, essentially for DMD, you want this to go to your skeletal muscle as well as your heart, right? And then how we can minimize some of the side effects, especially getting into liver or other tissues like we see in our intravenous gene therapies. And the intrathecal delivery was hypothesized as a way to address this challenge by testing a dose that is efficient enough to get into the muscles, but also skips the liver and other organs that we talk about from a safety or a side effect standpoint. As you could see here, you're seeing the darker staining on the muscle biopsy sections in the animals to indicate more protein expression through the intrathecal delivery compared to the intravenous delivery. And then you also see that the expression in the liver is sort of lower compared to the intravenous delivery. So that was just any sort of, just a gene that expresses a protein. We're just looking at intravenous route versus the intrathecal route. So this is the preclinical testing in animals to look at the amount of microdystrophin expression that's being made by this specific microdystrophin from the Insmed, which is INS1201. So the first column on the left shows the one that up here is the untreated DMD mice, and then you could see these different columns with different dose ranges that was studied in the mice, and the column 6 is your normal mice. And then if you could see these different rows, the first row is the what we call a H&E stain, just to look at the structure of the muscle itself. The second row kind of again gives you the picture a little bit more clearly. And the third row is a stain, it's a protein stain. And then the fourth row here is actually the microdystrophin, microdystrophin positive fibers in these mice. As you could see here, as you look at these different doses, there's a dose dependent response to the intrathecal delivery here. And that also, you can see the normal mice, the normal mice muscle tissue right here. And this is your untreated DMD mice. You see a lot of these, the muscle fibers don't look round, and then you see a lot of this purple. We call them nucleus outside of the muscles. Again, this is kind of tells you there is some muscle degeneration and fibrosis. And if you look at the person microdystrophin positive muscle fibers here, you could see there's a dose dependent response as you increase the dose, as you see over these two to 5 columns. And then here, yes, there is some, there is microdystrophin expression if you do this intrathecal route. And the next question is, how does it carries through your muscle function, right? So they checked a couple of exercises in the mice to look at how does it translate into the function. Now here again, you could see similarly, this is the green bars is the normal mice, and then you have different, and then the blue is the untreated mice, and you have the different doses that's being studied, that was studied in this animals. And as you see, there is the dose-dependent improvement in the functions that they tested in these mice. So one thing I just want to make a call out here is these are animal studies, right? These are preclinical studies, and we don't know how this is going to translate into humans. However, in a lot of times we do rely on the preclinical data to decide, to support our clinical trial to check in the humans and the boys with DMD. So just to say again, these are animals for animals, and then we don't know the effect is going to be exactly the same in the humans, but that's what the trials are being designed for. Like the same set of experiments were done 1 year after the administration of intrathecal INS1201. And again, you could see in these figures, there is a dose-dependent response. Here, especially, if you look at the dystrophin positive here are the untreated DMD mice in row one, and then these different doses, you see the more green staining, meaning we have more dystrophin, microdystrophin, and you can compare that with normal DMD mice. Again, there is the dose response of microdystrophin expression, and also similarly the effect of integrity to the muscle tissue. This is 1 year after the intrathecal administration. Now, these same preclinical studies where the DMD mice were dosed, there was also a dose-dependent increase in the microdystrophin protein found in the heart or your cardiac muscle. Again, it did shows that it did reach the cardiac muscle through, even though it was an intrathecal route. Again, I want to highlight this is a preclinical result. I wouldn't make so much conclusions in how this is going to translate into humans. But I think the hypothesis that the intrathecal delivery, the delivery through your spinal space, might target both skeletal and cardiac muscle is there, and more clinical trials are being done to validate that. And I think this kind of gave Insmed the validity to try this dosing strategy in the clinical trials to bring it to the clinic. I think this is mainly to just, when you talk about these different gene therapy, these different routes, we also want to know what we have seen from a safety standpoint in the animal studies, right? In the preclinical, what we call toxicology, which is a safety evaluation in two different animal species. They were done in both non-human primates and mice. And this was INS1201 was well tolerated without any observed adverse events across the range of doses that were studied. So tolerance in mice was based on daily observations of the behavior, their appearance, any signs of distress, tremors, any tail movement anomalies, and then the way they consume food. There are several measures that you look at, you observe in the animals to look at the safety. I think the other thing to highlight here was also there was a dose-related consistency in the biodistribution when you look at the mouse and the non-human primate studies. What that means is when crossing from one species to a different species, there was both a degree of predictability by the dose for the amount of expression, and there was evidence for expression in both skeletal and cardiac muscle, with limited to no non-muscle expression. Again, talking about expression in the liver and other tissues that you don't want this virus or the protein to go to. Again, these results strengthen the rationale for them for the underlying investigational dosing strategy for the ASCEND study and support the next step of testing this hypothesis in the clinics in boys with DMD to determine the safety as well as the efficacy. All right, just to kind of sum up, the intrathecal, the preclinical results from the animal studies with intrathecal INS1201 yielded microdystrophin expression that corresponded to the impact of the muscle tissue and impact of the muscle force and strength studied in the mice. Now, it also led to some microdystrophin expression in the cardiac muscle, the heart muscle, and those are also still seen approximately 1 year after the dosing as well. And there was no observed adverse events at the studied doses in the preclinical. Now, taken together, these preclinical results supported the continued exploration of intrathecal administration in humans with the range of doses that was tested in mice, right? And based on the doses, they studied 4 doses as we saw. Based on those the doses. Now, they chose the 2 highest doses from the mouse model to be studied in humans, in boys with Duchenne muscular dystrophy. And these are, like I said before, fixed doses. These are not weight-based dose. Fixed doses, they are as much as tenfold lower than what would be necessary for a weight-based dosing delivered intravenously. So as you could see here, this is the 2 fixed doses in 2 age groups that are being studied in the phase 1 trial, which we'll talk about in a minute. 2 to less than 3 years, and then 3 to less than 5-year-olds. And then these 2 different dose levels, 5 times 10 to the power of 14 and 10 times 10 to the power of 14 vector genomes. I think this is something coming from Insmed. I just wanted to emphasize. It's important to note that Insmed has worked on optimizing manufacturing and the analytical processes to help enable the predictability of a consistently high accuracy and high precision consistency. I think one of the things, the manufacturing is important for any microdystrophin gene therapy, AAV-based gene therapy, anything to minimize the safety. And I think this is just to highlight that Insmed has optimized their manufacturing process as well. All right, so we're going to move into now the phase 1 ASCEND study. It is a 2-part, multi-center phase 1 study. Now, phase 1 studies are typically done to evaluate the safety of a specific product, right? So this is a safety study. Safety is the primary objective of the study. And they're enrolling 2, 3, 4-year-old ambulatory boys with Duchenne muscular dystrophy. It is an open label, it's phase 1, right? So there's no placebo involved. Like I said before, it is the primary objective was to evaluate the safety and tolerability of a single dose of intrathecal delivery of INS1201. Now, there is, as any other studies, there is oversight by an independent data safety monitoring committee. This is a group of experts that are not involved in the clinical trial, that has nothing to do with the company itself. They are independently evaluating the safety of these boys as we get more data, as we enroll boys. And this is something standard that's done across all studies. And some of the tests that are being included, again, this is not new to the community, new to us, right? Like any other study, they get physical exams, blood draws, muscle biopsies, is a needle muscle biopsy, echocardiogram, so looking at an ultrasound of your heart, growth, development, as well as some questionnaires that's being completed. All right. So now this is how the enrollment is being planned. Since this is a phase 1 safety study, there's 2 parts to it. The very first part is enrolling 3 to less than 5-year-olds in we talked about these 2 different doses, right? The first dose is 5 times 10 to the power of 14, and the second dose is 10 times 10 to the power of 14. And then you have the 2 age groups, 3 to less than 5 and 2 to less than 3. So the first cohort, which is cohort 1, is the 3 to less than 5-year-olds. And then once, they were going to do 3 patients in each cohort. once the first three patients are enrolled, then there is a safety review meeting happening, and if everything looks okay, then there's a green light to proceed with these two cohorts. Cohort 2 here is the same 3 to less than 5, but at the higher dose, and then the 2 to less than year old, but on the lower dose. And then, once you dose them, 3 each, you're going to have the data safety committee review again, and they have to say, okay, to move to the high dose for this 2 to less than 3-year-olds. I hope this makes sense. I think it's pretty clear. Again, this is it's a sort of standard to look at two different doses. Just to emphasize here, both doses were effective in the preclinical studies, and there were no safety events in the preclinicals in the animal studies. Of course, it doesn't directly translate into humans, but if you want to kind of compare, that's how these two doses were selected, like I said before. And I think as of now, this cohort 1 has been fully enrolled. So let's talk about some high-level inclusion criteria for the study. So to participate in the study, of course, they have to be males, and it has to be 2 to less than 5 years of age with definitive diagnosis of Duchenne muscular dystrophy based on genetic testing with variants in the mutations in the DMD gene, including exons 18 to 58. As you can see here, the exons before 18, exons after 58 are excluded. I just want to highlight that here. And of course, the boys should be able to cooperate and perform motor assessments. And they should be able to walk unassisted for at least 10 meters. So here, again, this is the primary endpoint here is safety and tolerability, right? And after the dosing, they're being followed for 2 years of initial monitoring, and then optional extended safety study for follow-up up to 5 years, totaling of 5 years. Here, there is your pre-treatment period. It could be up to 46 days. This is your screening period, and this is where you also begin the immunosuppressive regimen that's being used here is the prednisone or steroids at day -1. And then you have the treatment, which is intrathecal injection. And then there is overnight stay for monitoring after the injection. And then there is 30 up to 100 weeks, there's follow-ups, right? That's where you have your blood draws. You have sometimes urine and all of the safety monitoring. If you need echo, EKGs, all of that happening in this follow-up period. And the prophylactic steroid that's being used is for 8 weeks. And then you have needle biopsies that are being done to look at the microdystrophin expression. And I also want to highlight, I think there's some remote and home health visit options available starting week 20 after the infusion. And then your separate follow-up study, which is the long-term safety follow-up. And then I think the study oversight, we talked about the independent data monitoring committee. It is usually the DMD drug development experts, and they provide review of data and advise the study progress. They're independent, meaning they're not actually conducting the studies, and they're not also employed by Insmed. They are the independent DMD experts. Thanks, Stephanie. Do you want to talk about this slide?
Sure. Thanks, Dr. Panda. So what we've learned from the community here is to help remove as many stressors as possible as it relates to participating for your kiddo in a clinical trial. And so we've teamed up with a support services concierge to remove barriers like distance and other to make sure that we can provide a comfortable experience for parents and caregivers and their children who may be interested in participating. And a lot of the features and benefits of those are listed here. And if you want to advance us one more slide forward, Dr. Panda. So today, as you mentioned, cohort 1 is complete, and we do have the active sites here are listed with a star across the US. So you can see all of the clinical trial sites Those that do not have a star will be supporting the study as well in due time. And if you want more information to find out more about the study and or from any of the centers directly, you can see on the right side of this slide here, you're always welcome to visit Insmed, medical information team. We're happy to provide information, but even more directly, you can go to ascenddmd.com and the QR code is there as well. I'm not sure how well that translates for those watching with your phone on a computer screen, but essentially going to ascenddmd.com to learn more about the trial and the sites that are open and enrolling and finding out who the best contacts at those sites would be, including yours, Dr. Panda.
Thank you. Yep. All right. So in summary, I think it's important to look at, in the preclinical animal models, the treatment with INS1201 yielded microdystrophin expression in both type of animals, both in mice and non-human primates, and functional improvements in muscle force and strength were seen also preclinically in the DMD mouse models. The distribution to the skeletal and cardiac muscle with low dystrophin in the liver of the animal models suggest further study of this intrathecal route was warranted. And I think together with these, they move forward with the approach of intrathecal route in the clinical study, which is ASCEND, the first in-human study evaluating the safety and tolerability of a single dose of intrathecal delivery of INS1201. And like I said, it's enrolling 2, 3, and 4-year-old ambulatory boys, and you heard about the study-related support services as well. Here is the contact information if you want to learn more about the studies, and always also talk to your care team about if you have, if you are interested in this or if you have more questions, they can always reach out to the company or to any of us involved in the study to discuss this more as well.